Mitotic chromosomal alignment is required for proper nuclear envelope reassembly. But what exactly does this mean? Before we can delve deeper into this mitotic chromosomal alignment, it is essential for us to further understand the cell cycle itself, but more specifically the M phase. So let's take a look at how cells make their way through mitosis. Mitosis begins with prophase, where all the chromosomes condense together but are not yet attached to any mitotic spindles. This spindle attachment will occur during prometaphase. This phase also includes the breakdown of the nuclear envelope. After this occurs, metaphase will begin and all chromosomes come together at the equator of the cell. Anaphase will follow soon after, allowing the chromosomes to begin to separate and move towards opposite ends of the cell, which leads into our final stage of mitosis, telophase, where the separated chromosomes will become enclosed in nuclear envelopes and decondense. But what would happen if the chromosomes were unable to come together at the equator of the cell during metaphase? The following image represents the KIF18A protein. This protein is a microtubule depolymerizing kinesin which has an effect on chromosomal congression, and it is primarily responsible for chromosomal movements during metaphase. One role of KIF18A is to promote proper attachment between kinetochores and microtubules. This means that a disruption of this protein will also lead to a disruption in the alignment of chromosomes at the equator of the cell. Deficiency in this protein has numerous effects on cellular behavior. For one, chromosomes are not brought to the equator in a uniform manner. Normal chromosomal movement is shown by the control cell in the top row, but when this protein is knocked down, the chromosomes are spread unevenly throughout the cell as shown in the bottom row. Surprisingly, this had no effect on the chromosomal copy number. It did, however, lead to two substantial defects in nuclear organization. For one, the nuclei of the KIF18A deficient cells are abnormally shaped. The nuclear envelope is normally round, but these cells are rigid and bent. This will primarily occur within the daughter cells. Secondly, these deficient cells will begin to form micronuclei as seen by the small dappy-stained dot to the left of the primary nuclei. But what is it that causes the formation of these micronuclei? During anaphase, the protein-deficient cells will have chromosomes that lag behind the others as they are being pulled apart, indicated by the arrows above. These chromosomes will arrive late to the nuclear envelope and a second micronuclei will form around it. But what are the results of the abnormalities of these cells? The above diagram indicates some major checkpoints in the cell cycle. The p53 checkpoint has been shown in the past to reduce cellular division of micronucleated cells. After depleting this checkpoint, proliferation increased substantially, showing that it is a major player in stunting cellular growth. Experimental analysis showed a 32.9% decrease in cellular division when the cells were deficient in KIF18A protein. But what could this mean for living specimens? KIF18A mutant mice were compared to the wild type to determine their differences. These mutant mice showed major defects including increased lethality prior to weaning and decreased body and growth rates. So what is the importance of chromosomal alignment? There are short-term consequences where the nuclear envelope and proliferation of daughter cells are greatly affected, and long-term consequences that affect the growth and overall fitness of the deficient animal. So what does this mean for our future goals? We will test KIF18A deficient cells for susceptibility to aneuploidy. This will help us determine if the protein affects the splitting of chromosomes during anaphase. We would also like to further analyze how this mutation could affect the fitness of living creatures, such as how the deficiency can affect stem cells, or how it can eventually lead to the growth of tumors.